right, so review for exam 3. We'll start in chapter 12, and you'll see we start with a short answer. Yeehaw! So the first one is to know the divisions of the nervous system, and I'll draw that out for you in this little clip coming up. All right, you should know then the definitions of sensory receptors. These are just our detectors for stimuli. Nerves are a bundle or a collection of axons in the peripheral nervous system. The ganglion is a cluster of nerve cell bodies found in the peripheral nervous system. And plexus is more or less compared to a superhighway of nerves that pass through a common region like your solar plexus. So remember the structure of the neurons, and you might want to review that particular structure. The structure of a neuron's prominent cell body with a visible nucleus. The nucleus has a dark spot in it called the nucleolus. Now the cell body is also known as the soma, and projecting off the cell body are cytoplasmic projections called dendrites, and there will usually be several hundred, if not thousands, of these dendrites projecting off the cell body, and usually one long extended cellular process called an axon. And it's the axon that is going to deliver the action potential to whatever the next structure is, be it a muscle cell, another neuron cell body, or its dendrites, or perhaps even a gland. So the cell body then, as we already talked about, has the big old nucleus in the middle. It's got extensive rough endoplasmic reticula, and that rough endoplasmic reticula, along with the ribosomes, are known as Nissl bodies. And you should also know there's plenty of microtubules and microfilaments present in the neuron. The axon hillock is the region of the cell body where the action potential will be generated. The presynaptic terminals are the ends of axons that are going to deliver the action potential. They'll possess neurotransmitters in them, and given the right circumstances, that's where we will begin our neurotransmission. Neurotransmitters are the chemicals inside the vesicles. Vesicles are storage compartments in the presynaptic terminal. The neurotransmitters will be packaged into them for eventual release. You always define sensory neurons as action potentials toward the central nervous system, and motor neurons as action potentials away from the central nervous system. The four major categories of neurons, we'll go ahead and just make that 3. We'll ignore the anaxonic and where they usually found. So the first shape we'll take care of is the bipolar shape. Remember what this is a cell body, and on either side of the cell body in one direction is a process that extends towards dendrites on the cell surface. On the body surface, or in the particular sensory organ surface. And the other extension from the cell body goes back towards the central nervous system. Bipolar cells are typically found in the special senses only. Now, unipolar cells have the cell body, but only have one process that extends from them, and that process splits, but it splits outside the cell body, and one side of that split goes towards the surface, and the other side of the split goes towards the central nervous system. These are your most common types of neurons that are found in the sensory system of the body. So they take care of our general senses like pressure, vibration, temperature, pain, and proprioception. Finally, the last type that we describe, probably the most of any, or the most common type found in the central nervous system, and those are the multipolar neurons that have extensive numbers of dendrites, to the point where you may even term them as dendritic trees, sticking off the soma and a long axon. And again, these are typically found entirely within the central nervous system. The next possible short answer is to know the 4 types of neuroglia that are found in the central nervous system, and then the 2 that are found in the peripheral nervous system. And for each, you should know a particular function associated with those. So I've got this listed out already for you. The 4 in the central nervous system are astrocytes, which make up the blood-brain barrier; ependymal cells, which are part of the choroid plexus that makes cerebrospinal fluid; microglia that serve as the immune system inside the brain; and the oligodendrocytes that are used to form myelin sheath around multiple axons within the central nervous system. Now, the 2 in the peripheral nervous system, the most typical neuroglial cell in the peripheral nervous system is the satellite cell, and it is simply a maid cell. It cleans up the environment, or perhaps you can think of it as a nurse cell where it keeps the environment clean and takes care of all the neurons that are present in its vicinity. The second type are similar to oligodendrocytes, but in the peripheral nervous system we call them Schwann cells. And Schwann cells can only wrap around one axon at a time. It can organize many axons, but they are not myelinated, they're just merely organized. Whereas an oligodendrocyte, remember, can wrap around many axons at the same time. Regardless, either one of these shells produce a insulating material made up mostly of phospholipid membrane, and that is called myelin. So that's the myelin sheath that is very high in phospholipids. Now, if we take a look a little more deeply at Schwann cells, we'll see that in between consecutive Schwann cells are bare spots. And those bare spots or gaps in between the Schwann cells are known as nodes of Ranvier. And here we have an incredibly high density of the sodium and potassium voltage activated gates that will allow passage of these ions according to their concentration gradients. This then results in a form of conduction called saltatory conduction that is like jumping conduction. It jumps from node to node, and this greatly speeds up the action potential along a myelinated axon up to the point of about 120 meters per second. White matter is just simply anything that's myelinated, which is pretty much just most axons. The axons that are myelinated appear glycine, so that's why we call them white, whereas groups of cell bodies and any axons that are not myelinated appear dark and dingy under a microscope slide. And so that would be called gray matter. Okay, so the resting membrane potential is made up of several factors. First, we have the notion that sodium is more highly concentrated outside the membrane of the axon, whereas potassium is more highly concentrated inside the membrane of the axon. Those are 2 major things. And then remember, no membrane is perfect, so we have the notion that we represent this with leakage channels. And we have leakage channels that allow sodium ions to leak inward towards the axon intracellular space, and then leakage channels that allow potassium to leak outward into the extracellular space. So that's another mechanism that forms the resting membrane potential. The fourth one is the sodium-potassium ATP pumps that are functioning to try to restore the normal amounts of sodium outside the axon and potassium inside the axon. And then finally, the last contribution to the resting membrane potential is the notion that there are many negative ions within the cytoplasm of the axon. And so this will give the inside a much larger negative potential compared to the outside. And so when we put these 5 things together, we get an average resting membrane potential for a typical neuron at minus 70 millivolts. So the next one comes to that guaranteed short answer, and I'm going to draw that out again for you, or at least paste in that portion of the video for you. Okay, so we're here to talk about the action potential. And on the left hand of the screen, I'm going to draw a graph. Okay, and on this graph, I'm going to begin at minus 70 millivolts. This is the only number I'll hold you accountable for. And so we'll start there, and excuse me, and we'll just have a nice straight line for a bit. And then what I'm going to do is I'm going to number that number 1. Okay, doesn't like to draw circles. And on this side of the screen, I'm going to put what happens at number 1. And so we're just going to put that it's simply the RMP, the resting membrane potential. Then what we're going to see is that we will start our stimulus, right? And we'll start rising. Oh my God. And here is my threshold. As I reach my threshold, then I will start opening sodium voltage activated gates, right? So I'm going to call this step 2. And then when I hit my threshold, I'm at step 3. All right, so step 2 is going to be sodium stimulus channels open. All right, and then step 3 is the threshold. All right, at that point then we will hit, like I said, our voltage gate, and we will have, let's see, let's do it in orange, and we will start seeing our voltage gate, the channels, the sodium portions begin rising fairly rapidly, and then we'll start to see a curve downward. So simultaneous with step 3 is step 4, and in step 4 I am seeing my voltage activated sodium gates open. Alrighty, and then we hit step 5 up here, which is the voltage at which they begin closing. And so I can just use ditto marks, which means copy the word above it. And instead of these guys opening, now we're seeing them close. Okay, step 6 will be over here, and it will just be right alongside step 5, pretty much simultaneous with it. Voltage activated potassium gates will open. All right, and they'll begin, that's why we see this downward descent. And we'll do green. Okay, and so we see this part of the action potential occurring, go below threshold, and then we hit the resting membrane potential. So towards the threshold down here, we have step 7. This is the voltage at which the potassium voltage activated gates will begin closing. And we'll hit step 8. I'll go ahead and keep it at green, and we'll descend under the resting membrane potential, and then eventually rise and get back to resting membrane potential. So step 8 is the part of the action potential called undershoot. And step 9, so let's draw that in here. Step 8, this position. Step 9, even though they are working the whole time, that this is going on, this is when we really notice them, and this will be the operation of the sodium potassium ATP pump. And that will eventually restore us to step 10, which is back to our resting membrane potential. There's a few other things I want you to know on here. And one of those things is the idea of what this phase is called. This is called depolarization on this side, where the signal is rapidly rising. I don't care that you know any voltages beyond the -70, but you should know that somewhere about right in here is 0. And so what we're doing is we're taking the signal from a very negative voltage, right, -70 millivolts down here, and we are having it rise towards 0, which is non-polarized. And so we are taking a very polar voltage going to a non-polar voltage. And it continues on to the top here. It then begins repolarizing. So this is called the repolarization phase of the action potential. And this bit under here that goes under the resting membrane potential, this is known as the hyperpolarization phase. Once again, what we're describing here is the action potential in all its glory, the generation of it. This signal will have many attributes that we'll discuss here in just a few minutes. You will have this on the exam. This is a 100% guaranteed question that you will have. I'll probably provide you the curve, but you'll have to type in or indicate where the various steps occur, and certainly have to be able to write out all the 10 steps. So I wish you good luck on that. Study this. Know it, know it, know it. Now, we talked about the sodium-potassium exchange pump. You don't need to know exactly what number of molecules are moved, just that it moves sodium molecules outward and potassium molecules inward. This is against their concentration gradient, so it requires ATP for this to occur, and that means that it's an active transport mechanism. What are some terms we can use to describe action potentials? We can say they do not change magnitudes. They are all or none. In other words, they either reach the potential and those voltage activated gates open, reach threshold and those voltage activated gates open, or it doesn't reach threshold and those sodium voltage activated gates will not open, and there is no action potential. So that's what we mean by all or none. You get it all or you get nothing. You gotta pass the line of the threshold. We also know that an action potential is self-propagating, and because of its very nature, it will employ positive feedback. One sodium channel will result in the opening of 2, which will open 4, which will open 8, and then 16, 32, and the number just multiplies very quickly. And that's exactly what positive feedback is. The absolute refractory period will be part of the action potential up until the point at which the action potential has decreased enough to go back below the resting membrane potential, which is the point at which hyperpolarization begins. Now, once we're under the resting membrane potential, when the potassium gates have closed and we've sunk a little too far negative, during that period of hyperpolarization, then we can have another action potential, but it takes more of a stimulus for that action potential to initiate. So that is called the relative refractory period. So absolute refractory period, you cannot have another action potential. Relative refractory period is the time in which you are in the undershoot, in hyperpolarization, when you could have an action potential develop if the stimulus is strong enough. The frequency of action potentials is just the number of action potentials per time. And so you can see that we can change this based on whether we can initiate one within that hyperpolarized state. If our stimulus isn't strong enough to do that, then the frequency will be slower. But if our stimulus is strong enough to make an action potential happen, even then when we are more negative during our hyperpolarization state, then our frequency will increase. And that will tell the brain something important, that there is a importance to this particular action potential because it is occurring more frequently. The diameters of axons vary all over the body. So there are type A fibers that are large, myelinated fibers. These are the ones that will have the saltatory conduction and conduct action potentials the very fastest possible way at up to 120 meters per second. The type B fibers are lightly myelinated, medium diameter fibers, and these are associated with the autonomic nervous system. Perhaps some of the more important information coming from the autonomic nervous system, you might think of things like sharp pain that would be conducted in type B, maybe type A fibers as well. The type C fibers are completely unmyelinated and are very small in diameter. An example of this would be conducting throbbing pain. They're slow to onset and they last for a long time. Now, you should be able to distinguish the effects of these different neurotransmitters. We discussed acetylcholine. Acetylcholine, remember, is the only neurotransmitter that we are worried about at the neuromuscular junction. It's also involved in alarm circuits in the brain. It will wake the brain up through activation of what's known as the reticular formation. So acetylcholine is very important in your wake portion of your sleep-wake cycle. And acetylcholine is also implicated in the effects of the drugs used for attention deficit hyperactivity disorder, or ADHD. Serotonin, whose abbreviation is 5-HT, is an incredibly important neurotransmitter for processes in the brain. One in particular whose outcome is a mood disorder. So if serotonin isn't functioning correctly in certain synapses, then you can get mood disorders. And a class of drugs called serotonin selective reuptake inhibitors, or SSRIs, are incredibly important for maintaining levels of serotonin in the synapse. So these drugs are the front line against major depression. Serotonin is also found in many areas of the body, many areas of the brain, but that's the main one I want you to remember. Dopamine's implicated in also many regions of the brain. The ones we're talking about are the major neurotransmitters. And dopamine's implicated in Parkinson's disease, so it has effects in movement. So in a certain region of the brain called the basal ganglia that you learned a little bit about in chapter 14, if we have neurons that have dopamine as their neurotransmitter in this region and they die off, then the result can be a form of Parkinson's. Dopamine is also important in reward circuitry, so part of the reward addiction pathway certainly utilizes dopamine. And then in schizophrenia, dopamine is implicated as well in quite a major way. GABA is your major inhibitory neurotransmitter in the brain, and all drugs for that combat anxiety have their effects on GABA receptors, and also any new anti-seizure drugs, anti-epileptic drugs are designed to work with GABA receptors. Epinephrine and norepinephrine are two neurotransmitters that are closely related, and they both have effects on mood as well. The first drugs that were designed against major depression worked on these particular neurotransmitters and their receptors, but they had large side effects associated with them. Epinephrine and norepinephrine are also known for their immense effects in the cardiovascular system. Finally, one that's left off, but that I do want you to know about, is glutamate. Glutamate is the major excitatory neurotransmitter in the body and has an enormous effect in memory circuits. All right, so finally, we end up with that the removal of these neurotransmitters from their pathways involves one of three ways, and that is enzymatic cleavage. So we talked about that for acetylcholine. So in this particular case, we had acetylcholinesterase that cleaved the acetyl group from the choline group. The choline group was taken back up by the presynaptic neuron, and the acetyl group just floated off and diffused away. Reuptake is another method used to clear neurotransmitters from the synaptic cleft, and the classic neurotransmitter that's removed in this way is serotonin, and serotonin is taken back up by the presynaptic axon terminal, repackaged into vesicles so that it can be re-released. And finally, diffusion is quite a common way. These neurotransmitters can simply just diffuse and find their way out of the synaptic cleft. So that covers chapter 12. Moving on to chapter 13. We had the spinal cord, in which we talked about our first discussion about the meningeal layers. We have the dura mater, which means tough mother. This is the most superficial of the three layers. And it is a very, very tough membrane. In the spinal cord, there is a space called the epidural space, and this can be utilized for injections to numb the lower limbs. Next, we have the arachnoid mater, and that means spidery. So you'll see spidery tendrils from the arachnoid mater more deep towards the pia mater, and this is where cerebrospinal fluid will flow. We also will see the subdural space, but we'll talk more about that when we get to the brain. And then the pia mater, which is pretty much adheres directly to the spinal cord tissue and is the deepest layer of the meninges in the spinal cord. I encourage you to take a look at a slice of the spinal cord and look at the arrangement of gray matter and white matter. You have the centralized gray matter butterfly pattern, and then the outer edges are the white matter axon tracts. We have an expansion in this gray matter that in the cervical area that accounts for the arms and neck and trunk, and then an expansion in the lumbar areas that accounts for the lower legs. The conus medullaris cauda equina. The conus medullaris is the end, the tip of the spinal cord, and the cauda equina are the nerves that emerge from this region around L1, L2, and the nerves that then proceed on down through the spinal canal. You should know there are 31 pairs of spinal nerves in the peripheral nervous system. And we won't worry about nuclei. You can scratch that one off. You should understand a little bit about dermatomes. Dermatomes, we can map those out, and specifically what that tells us is that there is a spinal nerve that is accountable for a particular region on your anterior and posterior surfaces, so that we can localize if you're having a particular issue in your skin or on your body wall. We can map it out to a particular cranial nerve, or at least to a particular set of 3 cranial nerves. Now, ventral roots exit from the gray matter of the spinal cord tissue, and those are all motor tracts, while the dorsal roots all contain sensory inputs. Prior to the dorsal root forming is the dorsal root ganglion, where the cell bodies of the unipolar axons that form the sensory components of the body, that is where they'll be located. The dorsal ramus will carry all information from and to the back. It's a fairly small set of nerves or axons, while the ventral ramus is much larger because it carries all the information from and to the rest of the body. The posterior gray horns can be divided up into containing somatic sensory nuclei or clumps of cell bodies, and visceral sensory nuclei or clumps of cell bodies. The anterior gray horns will possess the neurons for motor output, for somatic motor, and then the lateral gray horns will contain the neurons for the visceral motor outputs. We basically clump the three columns of white matter into whether they are anterior to the gray matter, lateral to the gray matter, or posterior to the gray matter. The major ascending pathways are the spinothalamic tract, and remember these are named for where they start and where they end. So this is a tract that starts in the spine, ends in the thalamus, and the thing you need to remember most about this is it carries pain and temperature information. The posterior column pathway brings information from your limbs, primarily information regarding two-point discrimination. And this is the idea of taking a compass with the 2 points that it has, putting them right next to each other, and pressing your skin, and then eventually spreading those points out further and further until you can detect when there are two distinct stimuli rather than just one. That's 2-point discrimination carried by the posterior column pathway. The spinocerebellar tracts carry proprioceptive information to the cerebellum. Sensory pathways, remember, all have 3 neurons with them. They have the primary neuron that takes the information from the body wall, from the skin surface, to the spinal cord. The secondary neuron that's completely within the central nervous system, from the spinal cord generally to the thalamus. And then the tertiary neuron that's completely within the brain that takes it from the thalamus, the information from the thalamus to that particular region in the somatosensory cortex that deals with it. Now, on the other hand, motor neurons all have at least 2 neurons. One is called the upper motor neuron. This is where the cell body is located in some region of the cortex. The axon will descend down into the spinal cord, and then in the anterior gray horn, you will have the beginnings of the lower motor neuron that projects through the ventral root into whatever particular skeletal muscle that it may be innervating. There are alterations to this when we talk about other types of pathways, but this is certainly the way it goes for the corticospinal pathway. So for a motor pathway, the main ones you need to know are the corticospinal, and these are movements of the body. The tectospinal, that is the startle reflex accommodations, so you don't want to be falling over, you want to keep everything in balance, and that's what the tectospinal does. The vestibulospinal, that controls balance coordination in the body, and reticulospinal, that deals with pain inhibition. Finally, in this chapter, you should know that reflex arcs are the very simplest types of nerve pathways, and you just have, for the very, very simplest ones, you have sensory input to motor output. But almost all of them involve a middle player, and that middle player is called an interneuron. All right, so we'll take this up in chapter 14 in the next review video.